Georgia. And years ago, in 2007, I had some people kept asking me for seed. I thought I need to do something. So I think in 2006, I think I gave out some seed to maybe about 5 or 6 people. In 2007, Mark contacted me and asked about getting some Ozark Chinquapin seed, and nobody could get them. They were just that hard to actually get them. And so in 2008, I sent him some seed, and he paid for his membership. And so he's been a member of the Ozark Chinquapin Foundation ever since 2008. I did not know at that time he was also the president of the Georgia chapter of the American Chestnut Foundation. And he's also been on the national board of the American Chestnut Foundation. And the gentleman has a wealth of knowledge, and it's really great to have this kind of expertise come here to our meeting and share a little bit about what he's learned and tell you a little bit about what some of the seed happened to do that I gave him. And so I'm going to go ahead and turn it around. Thank you very much.
Yeah, I'm happy to be here. You know, I've spent a lot of time actually in Arkansas. It's the first time I've ever been in Jasper, so I'm happy to be here. It's been a great conference. I hope to come back. So I'm with the Georgia chapter, although I'm not originally from Georgia, because I actually grew up in the Midwest in Iowa, which is well outside of the range of the American chestnut tree and obviously the Ozark Chinquapin. Although I do have southern roots because my mom's side of the family is from Arkansas, around, I guess, Walnut Ridge, Newport, that part of the state over there. So I can remember many summers coming down. My grandparents had a farm, Walnut Ridge, Lawrence County, I think that's right about in there. We'd come down, and if you're familiar with that part of the state, I'd go swimming Lake Charles, fishing in Shirey Bay, Horseshoe Bay, Horn Bay, right there on the Black River. So, growing up, I never heard about the story of the American chestnut, or even being down here on the farm, I never heard about the Ozark Chinquapin tree. Of course, I was probably, late 70s and 80s. Most of the Chinquapins had been eliminated pretty much, by then anyway. And of course, their farm is in this region, the Delta region. And if you saw on those range maps, you never see Chinquapin, shown here in this Delta region. Although, right through here, there's an interesting geological feature, Crowley's Ridge. And it's only about, I guess, about 500 or 600 feet above sea level. But it's kind of dramatic. It rises out of that flat, delta. I can remember as a kid, driving from the farm over here to Memphis and see Crowley's Ridge and go, wow, what a big old mountain it is. But so I'm kind of curious. Has anybody ever discovered Chinquapin on top of Crowley's Ridge?
I haven't read it, but a friend of mine saw a report from a Spaniard in the 1600s describing them on Crowley's Ridge.
Okay.
Near the Blue Hill.
Okay. So probably the newer range maps are like yes, it's really not Chinquapin anywhere in the Delta region here. So I didn't learn about the American chestnut until I moved to Georgia, and it was actually an article in the Atlanta paper describing the story of the chestnut. And I got doing a little bit of research and found out how important chestnut was to wildlife, to people, and then of course the science behind chestnut breeding was really fascinating. And so I became involved back, I think 1999, when I joined the Chestnut Foundation. And kind of through my work with them, I learned about the story of the Ozark Chinquapin, which is pretty much the same, but we're a bit wildlife to people. And so we formed a chapter here in Georgia in 2005. Right, so let's see. And this is when I was kind of researching and interested in chestnut. You go back and look at historical photos of chestnut. And this is an example of what used to be. You see the potential, the genetic potential of the tree growing in the right place. This is in the Smoky Mountains in Tennessee. This is what it used to be. And this is not even the largest by far the largest chestnut that ever existed. I think the largest one I've ever heard of was a 17-foot diameter chestnut in North Carolina. But that was a stump. So that was probably measured, just a couple feet off the ground. But still, DBH, that probably would have been a 15-foot DBH tree. So, it was a massive tree. So that's what it used to be with chestnut. And you saw that other picture Steve showed, massive chestnut trees. So this is about 10 years ago. This is actually me next to a chestnut tree in Iowa. And this is actually in the cemetery where a lot of my forefathers are buried there. So as a kid, growing up, I've been to that cemetery many times, but I never knew the story of the chestnut. Never paid attention to these trees. And one day I was there and said, hey, that's a chestnut tree. And there's actually 3 of them there. This is the largest one. So, if I do that, it's about 6 feet. So, DBH, I'd say 4.5 feet, maybe a little bit more. So not the largest chestnut tree in America, but that's out in Oregon. But still, probably one of the larger ones in North America currently. Okay, so we heard the first chestnut blight is Phytophthora, and Phytophthora is the genus. There's many species of Phytophthora that are pathogenic. Phytophthora cinnamomi seems to be the most kind of pathogenic. So when people talk about it, Phytophthora cinnamomi, that's kind of the one that they refer to. So this is kind of a map distribution of Phytophthora. The clear circles are 19th century reports, and I think right here in Savannah, Georgia, I think the port there is where it came in, started. You see over here in the Chinquapin range, it's the square, which is 20th century reports. So I don't know, does that kind of jive with what you've seen in the past?
I'm sorry, what now?
The 20th century. So you don't really see, you kind of see the progression of Phytophthora from this is from this source. So they believe Phytophthora came in at the port of Savannah. You see these white circles. These are 19th century reports of Phytophthora. You kind of see it spreading. And then the squares are 20th century. So you see right over here in this range, you only see 20th century reports of Phytophthora. And so, you'll see in the range maps that chestnut doesn't really kind of grow down here, and it's probably because Phytophthora came in and eliminated, the chestnuts in that kind of, this is called the Piedmont, kind of through here, it's called the Piedmont range. So chestnut blight, we've seen, that's really the second chestnut blight. That's Cryphanegria. Okay, you see the orange cankers. Let's see. All right, so let me give a background on THCF. So their official position now for about 2 years is called breeder. That stands for breeding, biotech, and biocontrol. So biocontrol, let me talk about that first. That's hypovirulence. And hypovirulence, I guess in simple terms, it's a virus that makes that blight sick. And then the tree, if the blight's sick, the tree can heal, kind of marshal its own resources. And it works. Hypovirulence works. We see it in, I guess in Europe. You see a lot, it works in Europe. Why? Why does it work there and not so much in America? It's kind of an open question. In America, there's different strains. The fungus, there's different strains. The virus can be incompatible. It's a complex question. It might work. The range for Chinquapin is quite a bit smaller. So the people I've talked to, general consensus is that hypovirulence is probably not going to be available as a tool for chestnut research or for restoration. Okay, breeding. So this is a pretty dense slide. If you've seen this before, you probably push the sleep. probably seen this 1,000 times. So this is the traditional kind of a plant breeding. Okay, so the idea, in general terms, is to move the genes from a Chinese tree into an American tree, and then ultimately produce a tree that has the blight resistance, okay, the Chinese parent, but that looks and acts like an American tree. That's the overall scheme. I guess it was back in the 30s, 40s, 50s. Two main organizations doing chestnut research would be USDA and Connecticut, what is it called, Connecticut
Research.
Connecticut Research Station. They gave up. They were working with trees pretty much in this generation right here. And this scheme, I guess, see how much I want to talk about this. So really the first step in breeding is cross the Chinese with pure Chinese and pure American. And you get what's called F1, and that stands for filial. The word filial, first filial generation, and it's going to be on average about 50% American genetics. Then the next, after you get the F1 generation, the next 3 steps are to produce a tree that increases the percent of American genetics. Okay, so you take F1 and you back cross it with the pure American 3 times, you get about 94% American tree. And at that point, the breeding program, according to the way it was set up, is stopped, and you do what's called an intercross. Okay, so the next 2 steps here, the percent stays the same. Okay, 15, 16. But this intercross, it kind of amplifies the resistance of the tree. Okay, so why do we intercross? Like, why don't we keep on going? Why not go up to a B6, get up to a 99% tree? Well, we could. There's nothing to prevent that, but it's logistics, time and money. Okay, so intercrossing has to deal with something called zygosity. And I'm not a geneticist. I'm not a tree geneticist or tree pathologist. So any scientists here can kind of help me out with this if you need. So producing a hybrid tree at F1 to a Chinese pure American, the best you'll ever get, and let me just explain this. These are chromosomes, okay, and what's in red is a gene. So, and there's 2 chromosomes because chestnut is what's called a diploid organism, because the gene's parallel. Human being is a diploid organism. So, this is just a simple example, but hemizygous would have 1 gene on one of the chromosomes. Okay, and the best you could ever get in an F1 generation is a heterozygous tree. You have the gene you want, and then possibly what's called an allele. It's a similar gene, but it doesn't have maybe the exact functionality. So if you got the gene from Chinese chestnut you wanted, and American had a similar one, it would be heterozygous. Okay, that's the best you can do from a hybrid. And then, so why do you intercross? You want basically both chromosomes to inherit the gene, same position, because that will sort of amplify the trait. Of course, the trait we want is resistance to life. A heterozygous or a hemizygous tree, will not have full expression of that resistance. Okay, and then biotechnology is the other B. That's a very broad topic. Transgenic trees, that tends to be, there's a lot of talk about transgenics. That's kind of the sixth topic. But really, THCF is embracing really the full, in the broadest sense, biotechnology, so not just transgenics. But we have our transgenic experts here today too. So this is just an example of this is a small spin assay. So you can see in this assay, pure American, pure Chinese, and two transgenics. The pure American basically died. Chinese, it healed itself somewhat. But these transgenic trees, really healed it well. They don't show anything like Chinese or pure Americans. And what a transgene is a gene from a completely different species. So a cisgene is a gene from like a Chinese, a similar organism. A transgene, happens to be, in this case, from meat. Could have been from a frog. People think transgenes, are like brain trees. If we use a frog gene, well, people really freak out then. So that does potentially, not only for American chestnut, but obviously for Ozark Chinquapin as well. So that's kind of the background of what national, the program is about. So in Georgia, we generally follow that. Okay. So what we do is a lot of what you do here. We're looking for native trees, pure Americans. All right, we're going to get pollen, possibly nuts. Put it in the breeding program. Then evaluate these trees, for resistance. Okay, this is kind of interesting. So this was produced by a gentleman from the DNR in Georgia. He took data from land claim records. So wherever it mentioned a chestnut tree, okay, he plotted that. And where you see red, just means there's missing data. The green shows where the tree was. So red doesn't mean the chestnut didn't grow there. It just means he didn't have the data. In fact, there's a whole chunk right here that he didn't have data for it. So wherever, like this land claim record, maybe mentioned a tree, here's a marker tree. So he plotted on the land claim records where chestnut grew historically. So that's the density map. That's the individual trees. And it overlaps pretty well. This is the geography towards it. So up here, that's the Blue Ridge Mountains. You kind of have what's called Ridge and Valley over here. Blue is Piedmont, it's called Piedmont region. Literally means the foot of the mountains. Up here, you're getting into the Cumberland Plateau, up here around Chattanooga. Okay, and then down here is green. This is the coastal plain. And so keep that in mind when you back up. You see where chestnut used to grow historically with the geography of Georgia. And you see that, historically chestnuts didn't really grow too well here in the plain. Although here's savanna down here. And that's where Phytophthora supposedly came into the U.S. So it could be on that data that Phytophthora had already eliminated trees, down in this part of Georgia by the time that data was collected. But, otherwise, it grows pretty well. That's what we find. When we go out in the woods and look at the trees, that's generally where we find the trees, is in this part of Georgia.